Okay guys, in this particular lesson what we're going to talk about is cellular respiration. So let's talk about that aerobic cellular respiration. We know that aerobic is for when there is oxygen, and in this case what happens is that the pyruvic acid, the pyruvate, is going to move into the mitochondria. So mitochondria is where the majority of all of that energy is going to be generated. The two steps that are going to occur there are the citric acid cycle, or Krebs cycle, and the electron transport chain. So when oxygen is available, all right, this up here, what we're looking at, that's that glycolysis. Remember, the first step that's going to occur in either one. And what's actually going to happen now is that we're going to move through a whole series of processes here in the mitochondria. That's what this image is right here. This shape is supposed to be the mitochondria. So what happens is that the pyruvate is actually used. One of these carbons is going to drop off as CO2, leaving two behind, but it's going to have a coenzyme. And remember, we said that coenzymes were there to help make sure that the reactions take place that need to occur. So that coenzyme is going to make sure that this acetyl molecule will bond with the oxaloacetic acid in the citric acid cycle, right, in this next reaction. So the citric acid cycle begins, and then the electron transport chain will follow. And our waste product is going to be water and carbon dioxide. But we're going to get a pretty decent amount of energy at this point, 36 ATP total from the citric acid cycle and electron transport chain. So let's talk about that citric acid cycle. Like I started to show you, that 2-carbon acetyl-CoA combines with a 4-carbon oxaloacetic acid to form the 6-carbon citric acid. And you'll see that this is where it's getting its name from, that 6-carbon molecule, the citric acid, hence the citric acid cycle. And the whole purpose of it is it's a cycle, so what you end with is what you begin with, and it's just going to constantly regenerate the oxaloacetic acid. And the oxaloacetic acid is going to bond with the next acetyl-CoA, and the cycle is going to continue over and over and over again, and it's just always recycling what its waste product is. What you get out of the citric acid cycle is ATP, NADH, some hydrogen ions, FADH2, and carbon dioxide. The NADH and the FADH2, those will move on to the electron transport chain, the next step in the process. And so long as oxygen and pyruvic acid are there, the citric acid cycle can continue. When you run out of one or the other, obviously it can't go on. So what happens, here's this kind of a close-up of that citric acid cycle. What actually happens is that, as we started to see, we've got the pyruvic acid from glycolysis. One of the carbons leaves as a carbon dioxide molecule. NAD comes in and forms an NADH molecule, and you're left with acetic acid. Coenzyme A is going to come in and is going to attach to the acetyl molecule, right, to the acetic acid, and what you're going to have is these 2 carbons joining the 4-carbon oxaloacetic acid, and what you'll end up with is 6-carbon citric acid. And then basically what's happening is that the 6-carbon molecule is going through a series of reactions as it moves through the cycle. Oftentimes it's losing a carbon molecule in the form of carbon dioxide. Sometimes it's giving off electrons and hydrogens for the NADH molecules to be formed. And ultimately what you wind up getting is that you have 1, 2, 3 more NADHs. You get 1, 2 carbon dioxides. You're getting 1 ATP molecule and 1 FADH2 molecule. in the citric acid cycle. And you can see each time the carbon dioxide is lost, our 6 carbon becomes a 5 carbon, the 5 carbon becomes a 4 carbon. And then really from here on out, all we're doing is kind of rearranging the 4 carbon molecule over and over and over again until ultimately it's back to that oxaloacetic acid that can move through in the next turn of the citric acid cycle. Just another picture showing you pretty much the exact same thing. And then the electron transport chain, which is the final ending place for aerobic cellular respiration. What happens is that NADH and FADH2 from our previous reactions are going to move into the ETC. They're carrying electrons, carrying all of that temporary energy so that the real form of energy, that ATP, can be generated. And all of these different reactions are going to happen in the cristae, the folds of the mitochondria, so that we can generate plenty of energy. And as you'll see here, this is where that water is produced as our waste product. And what happens is that energy gets transferred from the electrons that are in the NADH and FADH2, and it gets transferred to the ATP so that the ATP can be created. In order for that to happen, we have another enzyme. There's always enzymes that play roles. In this case, ATP synthase. Well, that should make sense. ATP, we know what that is. Synthase, well, ACE tells us that it's an enzyme. And synth, well, kind of sounds like synthesis, to make. So this is an enzyme that makes ATP. Well, that makes sense. And what ATP synthase does is that it makes sure that ADP is phosphorylated to become ATP, so the diphosphate becomes a triphosphate in the reaction. And then the oxygen, which is what you breathe in, gets the hydrogen ions and electrons in the end product and turns into water molecules. So just like this picture shows, we've got our NADH is going in, and we're carrying through a series of reactions, and all of our reactions are giving off a little bit of energy, a little bit of energy, a little bit of energy, and that energy is moving up to help create the ATP molecules that ATP synthase is playing a role in. And what we end with is going to move in with the oxygens to form our H2O. And this picture just kind of summarizes what I'm talking about with aerobic cellular respiration. It says, glycolysis, the 6-carbon sugar glucose is broken down into 2 3-carbon pyruvic acid molecules with a net gain of 2 ATP and the release of high-energy electrons. Citric acid cycle, the 3-carbon pyruvic acids generated by glycolysis enter the mitochondria. Each loses a carbon, generating CO2, and is combined with a coenzyme to form a 2-carbon acetyl coenzyme A, or acetyl-CoA. More high-energy electrons are released. Each acetyl-CoA combines with a 4-carbon oxaloacetic acid to form the 6-carbon citric acid, for which the cycle is named. For each citric acid, a series of reactions removes 2 carbons, generating 2 CO2s, synthesizes 1 ATP, and releases more high-energy electrons. The figure shows 2 ATP resulting directly from 2 turns of the cycle per glucose molecule that enters glycolysis. Because remember, I'm adding this in, sorry. Remember, when glucose splits into 2 pyruvic acids, each pyruvic acid has to have its turn in the citric acid cycle, so it really occurs twice for every 1 glucose. And then it says electron transport chain. The high energy electrons still contain most of the chemical energy of the original glucose molecule. Special carrier molecules bring the high energy electrons to a series of enzymes that convert much of the remaining energy to more ATP molecules. The other products are heat and water. The requirement of oxygen in this last step is why the overall process is called aerobic cellular respiration. Then we can talk about how these carbohydrates can be stored. I'm sure you guys have heard about the idea that what you don't use, you store. And sometimes there's excess glucose that your body doesn't send through cellular respiration, doesn't use immediately. And what actually happens is that instead of being a catabolic reaction, which is what cellular respiration is breaking down the glucose molecule, it enters an anabolic pathway where the glucose is built up, is combined with other glucose molecules to become a larger molecule. And that larger molecule is glycogen. So what happens is that after you eat, when your blood glucose is high, right, and you don't need all of the glucose that is in your blood system, the liver stores it as glycogen, stores it as a polysaccharide. So your monosaccharide glucose gets stored as a polysaccharide glycogen. and it's typically stored in the liver and in the muscle cells.